The following scenario is hypothetical and presented for educational and entertainment purposes. The first child did not survive. History remembers turning points as clean moments. A door opening, a discovery applauded, a crowd realizing the future has changed. But in this world, the moment that should have started vaccination collapses quietly inside a small rural clinic. The experimental protection against smallpox fails. The virus spreads through the boy's bloodstream exactly as it had through millions before him. Fever climbs, pustules bloom across the skin, and within weeks, the child is dead. In the original timeline, immunity began with a crude but brilliant biological insight. Exposure to a weaker relative of a virus could train the immune system to recognize the lethal one. It was the earliest practical manipulation of adaptive immunity. When antigens entered the body in controlled doses, B cells learned to manufacture antibodies. Memory cells formed. The immune system rehearsed the battle before the real invasion. Here, that rehearsal never proves itself. Scientists observing the failure reach a different conclusion. Instead of seeing cross-immunity, they see contamination, uncontrolled infection, and unacceptable mortality. The immune system appears too unpredictable to be safely trained. The concept of preventive inoculation is quietly labeled dangerous pseudoscience. Medicine therefore takes a different path. Without early vaccination success, research money shifts toward containment and treatment rather than prevention. Hospitals evolve into fortress-like isolation centers. Airflow engineering becomes the dominant medical innovation of the 19th century. Cities develop quarantine districts with complex filtration systems decades before antibiotics are even imagined. Immunology as a field develops slowly. Without a successful vaccine to inspire curiosity, the idea that the immune system can remember pathogens seems speculative rather than proven. Many physicians still believe disease spreads primarily through environmental imbalance rather than microscopic invaders. Ironically, the absence of vaccination accelerates certain technologies. Epidemiology becomes highly mathematical because predicting outbreaks becomes the only defense society has. Early statistical modeling emerges in government disease bureaus. Entire bureaucracies are built around surveillance of symptoms. But none of this stops smallpox. The virus thrives in dense cities of the Industrial Revolution. Mortality rates remain terrifyingly high. Entire neighborhoods are periodically emptied by outbreaks. Political leaders begin to view disease not just as a medical issue, but as a national security threat. And that is when control begins to replace medicine. Without vaccines, immunity becomes something rare and valuable. The small percentage of survivors who naturally recover from infection. These survivors carry antibodies in their blood. Their immune systems have already learned the fight that science could not teach. Governments start keeping records of them. At first, it seems practical. Survivors are recruited to care for patients since they cannot be infected again. Their blood is studied, transfused, and tested in crude experiments to extract protective factors scientists barely understand. But as outbreaks worsen, something darker emerges. Immunity becomes a biological resource. Military planners realize immune populations could operate in contaminated zones where others would die. Corporations begin funding research programs attempting to harvest protective compounds from survivor blood. The idea spreads quickly. If immunity cannot be manufactured, it must be acquired. And somewhere deep inside a government laboratory, a new proposal appears on a desk. Instead of preventing infection, what if infection itself could be controlled, not avoided, engineered? In laboratories that smell of antiseptic and burnt glassware, a quiet scientific revolution begins, not in the direction of prevention, but in the management of controlled suffering. If immunity cannot be taught to the immune system safely, researchers reason, then the only way forward is to choreograph the infection itself. Smallpox becomes less an enemy and more a volatile instrument. The biological reality of the virus is well understood by this point. Variola is devastating precisely because of its efficiency. Once inhaled, it infects the mucous membranes of the respiratory tract, replicates within lymph nodes, then spills into the bloodstream in a violent wave of viremia. Fever follows, then the characteristic rash as the virus attacks skin tissue and immune cells alike. But buried within the clinical records is a pattern physicians cannot ignore. Not every case is lethal. Some patients experience milder infections and survive. Their immune systems produce neutralizing antibodies and memory T cells capable of recognizing the virus forever afterward. In other words, nature occasionally performs the experiment that vaccination once promised, a survivable infection that results in permanent immunity. The challenge becomes engineering that survivable infection. Government-funded institutes begin searching for ways to weaken the virus without destroying its identity. Early attempts rely on temperature manipulation, chemical exposure, and animal passage experiments designed to force the virus to adapt to new environments. The goal is simple but terrifying. Create a strain dangerous enough to trigger immunity, but mild enough to avoid mass death. Yet without the historical precedent of vaccination success, public trust never develops. Instead of voluntary immunization campaigns, governments implement what they call controlled exposure programs. In densely populated cities where outbreaks appear inevitable, selected populations are deliberately infected with experimental viral strains under medical supervision. The logic is brutal, but mathematically sound. If 10% die but 90% survive immune, the next epidemic will claim far fewer victims. The first major trial occurs in a quarantined industrial district outside London. Thousands are exposed to a modified strain cultivated in laboratory animals. Physicians watch fever curves, measure lesion density, and track survival statistics like battlefield casualties. The results shock everyone. Mortality falls dramatically compared to wild infections. Survivors emerge immune, just as predicted. For the first time in this timeline, humanity has something resembling a vaccine, but it looks nothing like the clean preventative injection imagined in another world. It is infection management. The scientific community splits violently over the ethics. Some argue the program is a grotesque violation of medical morality. Others insist the mathematics of epidemics leaves no alternative. With smallpox capable of killing 30% of its victims, controlled exposure appears to be the lesser horror. But the scientific implications grow even larger. Researchers begin studying survivors more intensely. Blood samples reveal something remarkable. Specific proteins circulating in recovered patients neutralize viral particles directly. These molecules, antibodies, are the immune system's guided weapons. The discovery ignites a new field: passive immunity. If antibodies can be extracted from survivors, perhaps they can be transferred into vulnerable patients before infection overwhelms them. Laboratories start refining crude serum therapies, isolating protective compounds from survivor blood and injecting them into the sick. It works, sometimes. Enough to create an entirely new industry. Soon hospitals require a steady supply of immune donors. Corporations build blood extraction facilities. Survivors of controlled infections become medically valuable citizens, their immune systems functioning as living pharmaceutical factories. Governments issue immunity certificates. Employers prefer immune workers who can operate safely during outbreaks. Insurance companies offer better policies to those with proven antibodies. Entire economic structures reorganize around biological resilience. At first, it feels like progress. Smallpox no longer wipes out entire cities. Controlled exposure programs slowly expand across continents. Serum therapy reduces mortality further. Humanity appears to be adapting to the virus through brutal trial and error. But something else begins to shift beneath the surface. Because once immunity becomes measurable and profitable, someone inevitably asks a dangerous question. If antibodies are power, who should own them? Within a few decades, immunity stops being a medical condition and becomes infrastructure. The turning point arrives when laboratory technology improves enough to isolate antibodies with precision. with precision. Scientists learn that these proteins are not random defensive chemicals, but highly specific molecular structures. Each antibody binds to a unique fragment of the virus, an antigen, like a lock recognizing a single key. Once attached, the virus can no longer invade cells. Neutralization occurs at the molecular level. The implication is staggering. Immunity can now be quantified. Blood samples are analyzed for antibody titers, concentrations that reveal exactly how protected someone is. Governments begin maintaining national immunity registries. Every citizen undergoes periodic testing. A numerical score determines biological status: susceptible, partially immune, or fully immune. It starts as public health policy. During outbreaks, immune individuals are deployed as medical staff, emergency workers, and transport operators in contaminated zones. Because they cannot be reinfected, they become the backbone of an epidemic response. Entire sectors of society quietly reorganize around them. Then, corporations notice the data. Pharmaceutical companies realize antibodies can be purified, stabilized, and mass-produced from donor plasma. What began as crude serum therapy evolves into a highly engineered product. Each vial contains concentrated immune proteins capable of binding smallpox particles instantly after exposure. But unlike a vaccine, these treatments do not train the immune system. They borrow someone else's immunity. Protection lasts weeks, sometimes months, before the foreign antibodies degrade. The body never learns to produce its own, which means patients must return again and again for new doses, and suddenly immunity becomes a subscription. The wealthiest cities build massive plasma extraction centers where immune donors sit for hours while machines filter antibodies from their blood. At first, donors are volunteers, celebrated as heroes of public health, but demand rises faster than supply. Controlled exposure programs are expanded to produce more immune citizens. Certain populations, prisoners, debtors, and colonial laborers, are quietly recruited into exposure facilities where infection is induced under supervision. Survival means permanent employment as antibody donors. The mathematics of biology slowly merges with the mathematics of profit. Corporations patent antibody purification methods, storage compounds, and delivery systems. Governments sign exclusive contracts to secure supply for their populations. Pharmaceutical monopolies emerge whose entire business model depends on a simple biological fact: people must remain vulnerable in order to keep buying protection. A true vaccine would destroy the market overnight, so research into permanent immunity begins to disappear. Officially, scientists claim the immune system is too complex to safely train. Funding agencies prioritize antibody therapies instead. Medical journals publish endless studies refining dosage intervals, stabilizing proteins, and improving extraction efficiency. But inside certain research institutes, a few immunologists notice something deeply unsettling. The body already knows how to create permanent immunity. Survivors prove it every day. Their memory B cells can produce antibodies instantly whenever the virus returns, which means the immune system can be trained. It just has never been allowed to succeed. One young researcher studying immune memory begins reconstructing historical medical records from centuries earlier. Buried in fragments of forgotten correspondence and abandoned notebooks, she finds references to a crude agricultural experiment involving milkmaids and cowpox lesions. A theory emerges. If exposure to a related virus could trigger immunity without severe disease, it would eliminate the need for controlled infection and antibody markets entirely. It would create what humanity in another world would simply call a vaccine. She runs simulations of immune response pathways. The math works. Antigen similarity, epitope recognition, memory cell formation all align with modern immunology. But there is a problem. The global economy now depends on borrowed immunity. Governments rely on antibody monopolies to maintain order during outbreaks. Corporations worth trillions exist solely because people remain biologically dependent. A permanent vaccine would collapse the entire system overnight. And the moment her research proposal enters the network of institutional review boards, something unusual happens. The file disappears. Not rejected, erased. For several months, the researcher assumes it was an administrative error. Large research networks lose proposals all the time. Files misroute between departments, funding committees delay decisions, and sometimes projects simply vanish into the bureaucracy of modern science. At least that is the explanation she tells herself at first. But the deeper she digs, the stranger the absence becomes. There is no rejection notice, no archived review, no digital trace that the proposal ever existed. Yet fragments remain. Automated lab logs referencing antigenic similarity models, simulation outputs predicting immune memory formation, and a series of flagged security queries triggered the moment her data touched the National Biomedical Network. Someone saw it, and someone removed it. The scientific premise behind her work is deceptively simple. The human immune system recognizes pathogens through molecular fragments called epitopes. When B cells encounter these fragments, they generate antibodies and store the blueprint inside long-lived memory cells. If the same epitope appears again later, the immune response becomes nearly instantaneous. Smallpox survivors prove this process works flawlessly. Their immune systems retain that memory for life. But the virus responsible, variola, is brutally lethal during the first encounter. The entire problem of smallpox has always been about that first exposure. In this world, humanity solved it through controlled infection and borrowed antibodies. It works, but it traps society in a perpetual state of biological dependence. Her model proposes something radically different. A harmless relative of the virus could present similar epitopes to the immune system without triggering catastrophic disease. The body would recognize the fragments, build antibodies, and create memory cells before ever meeting the real pathogen. The immune system would rehearse the war without fighting the lethal battle. Permanent immunity. One exposure. Lifetime protection. The mathematics of epidemiology shift instantly under that scenario. If enough people develop immune memory, the virus loses hosts. Transmission chains collapse. Outbreaks stop, not because society controls infection, but because the virus cannot spread. Disease extinction becomes possible, and that is precisely why the idea is dangerous. The antibody economy has grown into the central pillar of global stability. Cities depend on scheduled immunity treatments during outbreak seasons. Military logistics rely on immune personnel. Pharmaceutical corporations control plasma infrastructure that stretches across continents. Entire political alliances exist around antibody supply agreements. If a permanent vaccine emerged, demand for those antibodies would evaporate within a decade. The plasma industry would collapse, controlled exposure facilities would close, governments would lose one of their most powerful tools for managing populations during epidemics. Biology would become autonomous again. That possibility terrifies institutions that have spent 2 centuries turning immunity into a controlled resource. The researcher eventually learns the truth through a quiet warning from a colleague in biosecurity oversight. Her proposal triggered a classification protocol known informally inside regulatory circles as immunological stability protection. Any research capable of eliminating dependence on antibody infrastructure is automatically sealed. Officially, the justification is societal stability. The transition away from antibody economies could trigger medical shortages, financial collapse, and public panic during the decades required to vaccinate the world. Unofficially, the reasoning is simpler. Power rarely dismantles systems that generate it. For a long time, she considers abandoning the work. Many scientists in her position would. Careers vanish easily in a world where research networks are tightly monitored and funding flows through centralized approval channels. But the biology refuses to leave her alone. Every time she examines immune cell data, the same truth stares back at her. The human body already contains the mechanism needed to defeat smallpox permanently. Memory cells, antigen recognition, epitope mimicry, The system is waiting for the correct signal. And somewhere in nature, that signal likely already exists, not in engineered laboratories, but in animals. She begins quietly studying viral relatives circulating among livestock and wildlife, searching for genetic cousins of variola with similar antigen structures but lower pathogenicity. It is painstaking work done off-network, using isolated equipment and physical notebooks instead of digital databases. Months pass. Then one night, while sequencing viral proteins from a rural cattle infection, the alignment algorithm reveals something extraordinary. A near-perfect epitope match, a virus that could teach the immune system to recognize smallpox without killing the patient. In another timeline, centuries earlier, this discovery would have ignited the birth of vaccination and transformed medicine into a science of prevention. Here it threatens to dismantle an entire civilization built on the business of survival. She stares at the data for a long time before shutting down the lab lights, because now the question is no longer scientific. If the first vaccine failed long ago and the world rebuilt itself around that failure, what happens when someone finally succeeds?